Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science Podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I am so happy to introduce a very special guest on the show. With us we have Dr. Rob Armstrong, who is the CEO of Artax Biopharma, a company advancing treatments for autoimmune diseases. Artax is specifically developing a class of oral small molecule drugs called Modulators, which are designed to modulate T-cell receptor signaling to restore immune balance without the immune suppression that often comes with current therapies. Dr. Armstrong became CEO of Artax Biopharma in January 2024 after previously serving as chairman of the board. He was also CEO, board member, and co-founder of Boston Pharmaceuticals. With over 20 years of R&D experience, Dr. Armstrong has led drug discovery and development programs across multiple therapeutic areas at companies including Amgen and Eli Lilly, where he held leadership roles in medicinal chemistry, translational medicine, and external R&D. Before joining industry, Dr. Armstrong was a tenured professor of chemistry and biochemistry at UCLA. Dr. Armstrong holds a Bachelor of Science from UC San Diego, a PhD from California State University, and was an NIH postdoctoral fellow at Harvard. He also advises and serves on the boards of several early-stage biotech companies. So to learn more about the work that Dr. Armstrong is leading and spearheading at Artax, let's turn to him now. Thank you so much for being on the show with us today, Dr. Armstrong.
Thank you very much, Ayesha.
I'd like to first learn more about your background in terms of actually transitioning, going from academia to industry, and specifically what ultimately led you to lead Artax Biopharma as its CEO, and what really excites you about the company's mission and goals.
Well, thank you very much for that question. I started my career in academia. I assumed I would end my career in academia. But given the research that we were doing in our labs, there was an opportunity to spin that out as a potential funded biotech. And so we in fact did that. And I got involved with some of the VCs that were funding the company, as well as some of the pharmas. And it became clear to me that there was a very exciting and interesting opportunity to be working not only in academia, but also in pharmaceutical and biotech industry. So we started a company called Ontogen in the early 90s. As many of the startups didn't actually pan out as we were hoping it would, but I certainly got to get to meet some of the leadership of some of the big pharmaceutical companies, and was asked if I would move over to Amgen at that time and start their small molecule program. At that point, they were mostly a secreted protein company. And I moved over there and ran the R&D for small molecules, started it up, and got some buildings built because they had no chemistry facilities. And the rest is history. That group was quite productive and continues to be so.
And so from setting up labs in industry to now CEO of Artax, how did that evolution arise, and what particularly drew you to Artax Biopharma?
So a couple of things. Number one is when I started out in industry, it became very clear to me that this is just a tremendous learning ground on how to actually do drug R&D. The level of expertise, the level of different disciplines that are necessary to put together even a decent shot on goal to go into the clinic, in particular with a new and novel mechanism, very exciting, but a great learning opportunity in the company. So that's what I did at Amgen. I then moved on to Lilly, as you pointed out. And once again, I used those opportunities to learn from all the groups that I interacted with, all the individuals, and just tremendous talent in these companies. So at that point, I still had this itch of continuing to be an entrepreneur, as I had been kind of coming out of academia. And so after a couple of years, I ended up getting involved with a number of VCs, both as an advisor or in investment committees and VCs. One of them was called Columbus Ventures, based out of Madrid and in Spain. And Columbus was one of the early funders, of Artax Pharmaceuticals. And so that's when I really got involved with the board. I really sat on that board, I think, for 11 years now. So been involved since the inception of the company, was the chairman, and then there was an opportunity early last year to become the CEO of the company. So that's kind of how I've been involved with them since the beginning. We've been elucidating some really complex biology that was fairly unknown when this company was founded. Balbino Alarcón, who was the scientific founder of the company, professor in Madrid, really has done a tremendous job to elucidate what I think is just amazingly elegant biology associated with the T cell receptor. And so we've been developing the science behind this. It's taken 10 years to really figure out exactly what was going on. We've had a shot on goal in the clinic early on with a molecule that didn't turn out to have the decent properties that we thought we could move forward. So we retooled that and put in a second molecule, and that's the topic of some of the recent clinical data that we've actually been publishing.
Yeah, that's really really great to hear how long you've been with the company. It's not just you were recruited externally. You were really at home in a way where you were serving on the board and you saw the whole evolution. So that's really nice. I'm sure that was a really nice transition to now leading the company to really great things. And so what do you say would be the core missions and goals of a company as we always talk about? So for Artax, what really is the focus?
Well, when we started out, I in some ways, I'm a sucker for novel new targets to work on. I'm also scared to death of them because it's always nice to have somebody ahead of you that can validate some of the biology around these targets. And then you can come in and try to improve on properties that they've identified up to that point. So what we set out to do originally, probably as always, evolves to what it really turned out to be later on. But what in the process of elucidating the biology around this target, we came to realize that this is just an absolutely unique adapter protein. It's called NCK, non-catalytic kinase, meaning it's not your standard kinase. It really is an adapter protein that serves many different functions, some of which are critical. So you can't really inhibit this protein as a whole. You couldn't do a degrader strategy to this because this would be a very it just wouldn't be a viable drug. So what we've done is figured out what part of NCK needs to be modulated without touching all the other functions which are critical to the cell of NIC. So it's taken us some time to figure that out. What makes it unique is that this is really a protein that amplifies a TCR signal. When an antigen binds to the TCR, it activates the TCR, obviously, and starts a cascade of events. And if you think about it, it's a very unique receptor because it has to accept both very strong avidity antigens as well as incredibly weak avidity antigens. For the weak ones, what it does, its function is really to amplify these so that you can start the immune cascade. For those that are really strong avidity, it really is not necessary. And so while our small molecule modulates and binds to it, it really isn't necessary for strong antigens, but it is for weak antigens. And that distinction is between autoantigens, which tend to be weak, and then pathogenic bacterial viral antigens, which seem to be very strong antigens. So uniquely, this approach allows us to start to differentiate between just knocking down the immune system or really modulating the immune system to focus only on autoantigens associated with autoimmune diseases while maintaining the safety that would otherwise be compromised with approaches that actually completely go after the immune system and suppress it globally instead of very exquisitely like we do. So we're looking at safety. That's a component of this. And this is important because some patients can't tolerate some of the drugs that actually would work, or they have to come off those drugs on holidays or otherwise, or some suffer from quite severe opportunistic infections associated with knocking down the immune system in those patients. So we're looking at that. That's one component: is safety, this differentiation, which we think is unique out there. So we're in some ways, the second part of it that we look at is that it's at the headwaters of the cascade of the immune system. What does that mean? It means that historical approaches to autoimmune diseases are looking for a cytokine that appears to be the most important cytokine for that particular disease. For some diseases like psoriasis, that tends to be pretty focused on IL-17, but in many diseases, they're much more heterogeneous in terms of the T helper cell population that is affected by that particular disease. So what it allows us to do by being at the headwaters is that we actually can cut across all the Th cell lines, so Th1, Th2, and Th17. So the advantage that has is it's as if we were multiple different drugs that are targeted to different autoimmune diseases. We believe theoretically, in theory, that, and we hope to prove, and we're starting to do that in the clinic, that this is applicable across all the disease states, autoimmune, that are affected by Th1, Th2, and Th17. So I'll give you an example. One is like atopic derm. I mean, Dupixent is a great drug. It really goes after Th2, Th17 cells, but it doesn't affect Th1. And yet it's very clear that in patients, in particular elderly patients, Th1 is actually is a player in the disease itself. So what we're looking at then is a potential to go after patients that aren't served by some of these other drugs, as well as to actually add on to some of those other therapies that already exist.
Wow, that's really wonderful to learn about really the elegant biology behind all of the signaling pathways. And Nick is a protein that I feel like we don't hear too much about, or maybe it was just it was just my ignorance. But I was reading up more about it, and yeah, like you said, it's so ubiquitous. It has your typical SH2, SH3 domains. It's kind of like a scaffolding protein, and it's so critical. And yet, the approach here, as I'm understanding, is just to modulate a specific domain, so to modulate that immune cascade. So, and again, like you said, I've worked in the space for my graduate research as well. And yes, the cytokines, they're always front and center, but this is a very unique approach, and it's very exciting to hear about it for sure.
And you bring up an interesting point on the domains. This has 4 domains. One of them is SH3.1.
Oh,
There's a very unique pocket there that we actually have a small molecule that binds to it, very potent, about 1 nanomolar. And what's interesting is that drugging SH3 domains historically has been incredibly difficult to differentiate between one or the other. The reason this works uniquely is it's just a different SH3 point domain. It has a different pocket. So we have tremendous selectivity towards this compared to any other SH3 domain. I don't think that, we're not proposing that we're an SH3 company that could provide a platform of drugs in that area, because I don't think we would ever get selectivity. But this turns out to be very exquisitely different. We never predicted this. It's just simply an accident of the R&D that we've done in elucidating really both with crystal structure as well as co-crystals of a drug analog to the site, that we really understand why it differentiates from all the other SH3 binding domains.
Yep. And when we talk about targeted therapies and all of that, this is exactly the kinds of approaches that that we indeed talk about. And so in terms of, I guess when we start talking about the indications and autoimmune diseases in general, I guess just stepping back a bit, in your opinion, what really are the current unmet needs as we talk about the unmet medical needs in autoimmune and inflammatory diseases? You mentioned a couple of them that Artax is focusing on with its pipeline. And so what are some of those indications that you're pursuing these modulators in, and what are the unmet needs that are driving really this approach?
Great question. I think sometimes we don't give ourselves as an industry and as academics working in this space the credit of the progress that we've made in generating drugs that actually are working for a large portion of patients across all diseases. You look at oncology and it's now down to the mutation in some cases, right? If you have that mutation, we have a drug for it. If you don't, we don't yet have a drug, but I assure you there are companies out there working on trying to get there. So it's just a snapshot in time as to how this progress has gone forward in terms of identifying drugs. So when you look at autoimmune diseases, I look at RA, for instance. I think there are 11 approved biologics and one small molecule. And yet there's a large proportion of the population that actually doesn't respond to these existing drugs. So we and others spend a lot of our time looking not at the generic unmet medical need of disease X, Y, or Z. It's really the fine tuning of which patients are not responding or unable to take those medications for a long period of time because they have some other comorbidity or compromising items that they have to deal with. So really, it's going after those populations that we think this drug actually can serve. So I can go through the whole slew of, it could be, it could be RA, it could be atopic derm, it could be psoriasis, it could be some of the GI autoimmune diseases, right? Rheumatology, autoimmune diseases, and every one of those has a drug that's in a time. If you look at the timelines, it's incrementally adding drugs that actually benefit patients. So that's where we find ourselves, is potentially adding on to other therapies because we don't think we have any reason not to be able to do so, but also not only just add on, but also serve those patients that actually do not respond to existing therapies. So RA, again, is a great example. There's a huge population of patients that are underserved, and there's some great drugs that actually work really well. So it's a matter of continually trying to go after the 100% of the patients that might have that disease. And it'll require sometimes more nuanced and more targeted approaches. And that's what we think this is.
And so, Nick, being such a ubiquitous protein and player in the immune cascade and in other signaling pathways, have you found that there are certain subsets of patients that benefit more from this approach with these small molecules, or is it more of a universal effect? Are patients, let's say, who are on other drugs, would they benefit from this as well, or is it just a certain subset, the ones that don't respond to the therapies already out there? Because as you mentioned, and rightfully so, those are the patients that must be the focus in terms of those unmet needs currently. So is this an approach that is ubiquitous or universal for all, really, or just for certain subsets?
No, actually, we think it's universal. And we think it's universal for the reason that we're going after all the T helper types, right? So we cut across those. So we don't think there's a subpopulation of patients that would benefit more from Nick modulation, for instance, than others. And to the second part, yes, we believe that it can be used in combination, and that would actually be beneficial for even existing therapies that might not cut across all the T cell types, but are quite effective at some of these. And if you look at some diseases like psoriasis is a good one. I mean, there are a lot of mechanisms that work in psoriasis. Some work even deeper than others, right? But the kinetics of the onset of the effect of the therapeutic agent oftentimes just depends on what cytokines involved, or is it a secondary event, et cetera, et cetera. So if you look at psoriasis, the onset with an anti-IL-17 is quick. Is a couple of weeks you start to see effects. But it turns out that some of the slower mechanisms like IL-23s take a lot longer, but the effect is deeper. So we see an opportunity to layer on top of these, both on the kinetic side as well as the depth of the pharmacodynamic effect. We think we can impact both sets, right? And we can have a positive outcome on both.
And you mentioned that, and also at the top of the show, I mentioned that there's some exciting new positive clinical trial data that the company recently announced. And so, could you share some of that data with us, and where that data and how you're building on that?
Sure. No, happy to do so. The again, new mechanism, first in man, first in class. So the fear you have, of course, is that you go in the clinic and you see absolutely nothing, right? So happened to me before, so and probably will happen again. So the at the time we did the study, we asked ourselves the question: what patient population should we go after? Which, because we have this plethora of autoimmune diseases to go after. At the time, we had 4-week safety data. So really, the question was, of all the diseases we can go after, which one would more likely to give us some readout, whether it's clinical and biomarkers, in a 4-week study that would start to give us some semblance of confidence that the biology that we're associating with this, we think the therapeutic effect that we're going to observe is actually real instead of not. And I might add that this is a pharmacologically silent drug if you don't have a disease. And what do I mean by that? In Phase I study, which we ran, which was a SAD/MAD study in healthy volunteers, we saw nothing, right? We saw no effect on any biomarker because these are healthy patients. So we're pharmacologically silent. So we went into psoriasis because in psoriasis, in 4 weeks, you can start to see biomarkers that are indicative of whether you see improvement in the disease or not. These are very well understood, very well studied. Pretty much every mechanism out there has done biomarker data in this space. We were working with Dr. Jim Kruger at the Rockefeller University. He's worked with numerous other biotechs and big pharmas with their different mechanisms. So he would be able to give us some semblance of where does this stack into all the others if the data is positive. So we ran the Phase I. It was very safe. It was like water. So I start worrying whether there will ever be an effect here or not. And we started our trial in psoriasis last year. It was a 30-patient trial, double-blinded, placebo-controlled, 2-to-1 randomization. And we saw the data early this year. And we were quite pleased to see that all the biomarkers that you would anticipate moving in a patient, as well as histological readout of these patients, were consistent with this being a biologically active molecule, and were consistent with the improvement of patients that were on this trial, statistically significant in many biomarkers that are relevant to the disease. And we even saw in moderate patients, because we ran a mild to moderate study, that in the moderate patients, we could see the PASI scores differentiating from placebo, not statistically significant, but didn't anticipate that in a 4-week study. What that did is it gave us tremendous confidence that this is really a mechanism that's relevant, that's biologically relevant to at least the disease psoriasis. This is Th17 cells. We're interested in going then into new indications. We're looking at atopic derm potentially. We're looking at rheumatoid arthritis, for instance, to go forward. And not surprisingly, those are Th1 and Th2 diseases, which is going to give us some data in patients of this concept of universality across the Th helper populations that we're looking at.
Fantastic, and congratulations on that data, and really you have something to build on there. And in terms of really taking these NIC modulators and being able to modulate this specific axis or this specific system or signaling pathway involving these NIC proteins, are there other diseases that these modulators could be extended to and expanded to?
Yeah, we've looked at that too. Besides all the kind of big autoimmune diseases, right, that we've discussed, it turns out there's some orphan diseases that are T cell specific, T cell modulation, T cell receptor driven, that we think this mechanism could potentially work in. So there's that. We have some intriguing data in IO models of cancer. This is not an oncolytic agent, won't have anything to do with the cancer itself, but we have some intriguing data showing that we can improve tumor kill and decrease any autoimmunity associated with IO AEs. And so we have one very interesting model where we can actually look at weak antigens, which would be the autoantigens, because some of the patients on, for instance, you know, PD-1, CTLA-4 antibodies develop, for instance, type 1 diabetes, which is an autoimmune disease, right? And so oftentimes they're treated with prednisone to try to address that part of the disease. But of course, when you do that, you shut down tumor kill and you see tumor growth initiate again. So we did a really interesting study with a model that actually had tumor. We were looking at both tumor and kind of the autoimmune component of it, and we can show that we affect the autoimmune very beautifully, but we don't affect the tumor, right? So even though it's not an oncolytic drug itself, it could potentially be given along with these IO therapies that would allow perhaps the therapy to be given much longer without some of this, RSAEs that you see in that space.
Absolutely. And it also brings to mind the tumor microenvironment and all of the complexities within that, with, in terms of immune cells and the composition of the microenvironment there, and to potentially modulate that.
So we're an autoimmune disease company,
Hmm.
so I don't mean to say that we're in
Yeah.
oncology too, but I'm just saying in response to your question, we have some allogeneic data that looks really interesting. So it's a matter of modulating the immune system for other modalities, other disease areas, et cetera, et cetera. So I think 10 years from now there'll be a lot of really interesting data, hopefully some drugs in this space that are chasing down some of these other disease areas.
And, in recent years, biologics have really blown up, and we've seen so many approvals um, of biologics in recent years. And so, how do you position yourself with small molecule drugs? They've been around forever. Um, are they a bit more traditional? Maybe people don't see them as exciting, but how do you position yourself within this really growing space where that is being increasingly dominated by biologics?
Again, it's a great question. And first of all, I didn't mention it. We're once a day oral, very small dose, so very convenient, right? The way I'd answer it is I think you're absolutely right that some of the major breakthroughs in autoimmune diseases really have come from antibodies. And again, it's these antibodies that are really focused on some cytokine that is kind of the bad actor of a specific disease. We said before, but maybe doesn't address all patients that have that disease or the disease as well as it could. So there's no doubt that that's been excellent. It'll continue to be great. And there are new antibodies coming out all the time. I think you'll see bispecifics playing a bigger role even in autoimmune disease, right? But the trend that also has been occurring over the last 5 years is those are validated mechanisms. So the small molecules have actually started to go after validated mechanisms that I think many of us 10 years ago thought would never be possible. But now we actually are seeing small molecules successfully moving through the clinic that basically are the same validated mechanism, but now you're just switching modality from an antibody to a small molecule. So I'll give examples. I mean, the J&J IL-23 oral peptide looks fantastic in atopic in psoriasis. I mean, it looks absolutely fantastic. So much so that I think it'll certainly be a big player in that disease going forward. You saw there's a molecule from Dice, IL-17 oral. Uh, Same thing. I mean, what you're seeing, I think, is uh, the idea that, and the second trend in parallel has been that peptides are all of a sudden oral drugs that we never really, other than a couple of exceptions going way back historically, now you're looking at oral drugs that are peptides. So I think what's interesting is that you have now peptides coming in, which are orally dosed, so they look more like a small molecule. They're certainly much smaller from a molecular weight than an antibody. So I think you're going to see a lot more shift in that direction of oral drugs that are going after validated mechanisms that have worked historically in with antibodies. I don't think it changes the fact that you're going to see antibodies continue. Again, bispecifics, I think, might up the game for historical antibodies, right? But I still think you're going to see a constant pressure to come in with and back up these, and it may take a decade in many cases. But certainly there've been some breakthroughs that I think, again, maybe going back 20 years ago, were maybe not envisioned as possibilities, and I think you're now seeing them occurring. So that's what I would say is I think patients at the end of the day, convenience of a small molecule, but if you get efficacy with a injectable, patients are going to, and they don't have any other recourse in that disease state. I think they all benefit. Everybody benefits from these modalities.
It's about everyone winning, and I think I really like your perspective and an approach in terms of you're here not just to compete or just look at that, really to build in this space, to layer and to really encompass all patients and to really help every single patient out there. I think that really stands out for me, at least from this conversation. So that's really, really wonderful. And I really respect and love that approach.
The goal for all of us, right, is to
Yeah.
serve 100% of that patient population. And we all will have different approaches. And the more impact you have, I think the harder it is to go after that last patient population. There's a reason they're not responding to current therapies, right? So to me, it's kind of a relentless attempt to try to go after that population that doesn't respond going forward. So.
And then there's always building, because then you have next gen drugs just to improve the current quote unquote model, and just to keep improving the existing classes of drugs that may be working well. So there's just so much scope and opportunity, and just to keep building. So.
And in oncology, the concept of polypharmacy, right, is well embedded. I mean, you take a cocktail drugs, it's a combination of this. I think you're about to see that movement, and you're already seeing it in autoimmune diseases. It's this combination of mechanisms that maybe is what's critical to really address these unmet medical needs that are a smaller part of the general population.
Indeed. And in terms of really the landscape of autoimmune diseases, as we've talked about biologics, and now small molecules are making a comeback in a different way. Again, the convenience of them cannot be understated or overstated. But what trends do you see in the space in the next coming years? Is it still going to be really targeted therapies, and like you mentioned in oncology, getting down to like the mutations and at that level? And so where do you see the space of autoimmune diseases heading in the next couple of years?
Again, by specifics, again, combination therapy. I think identifying new targets that there's a lot of work. There's some really interesting targets in phase 2, phase 3 right now that I think are going to start to show this more universal effect than we've seen before. I think signal 1 targets, both small molecules as well as biologics, in signal 1 autoimmune kind of cascade, I think have a huge potential to be broader than some of the more targeted historical approaches. So all this is playing out right now in real time. You're seeing clinical data popping up every single month in some of these mechanisms that are, I think, new and really novel, and I think change the landscape again, and hopefully again address efficacy to get higher efficacy for patients, and address these populations that are not that really don't have anything right now. So I think again, the beauty of this industry is just a plethora of ideas, approaches, modalities. Mechanisms, it's just a continuum in evolution of understanding how to address some of these diseases.
And what can we expect from Artax in the coming months and in the next couple of years, really contributing and leading in this space?
Well, I mean, what we're hoping to do is initiate a couple of new clinical trials going forward. Um, we right now are targeting RA and in atopic derm, but we're still evolving in our thought process of what to go after forward. I think these studies, once we actually complete them, would be reading out in roughly 2027. So we would have an idea of both impact on those disease states as well as this more general approach of universality of we can go after many disease states that are affected by the different Th cell helper cells.
Well, Dr. Armstrong, thank you so much for your time and insights today. It was so wonderful learning about your journey from academia to industry to entrepreneurship, and about the absolutely fantastic work that you're leading at Artax Biopharma. And we wish you all the best, and we're going to keep our eyes out for all of the latest developments, and really looking forward to seeing what's coming up from Artax. And thank you once again for your time.
Well, thank you too, but I also want to give credit to the team. Obviously, that's what it takes. We have some brilliant scientists and clinicians on board, and really also a very supportive board that has allowed us to take this forward. So thank you very much for the time. I really enjoyed it.
Likewise. All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.